Hello, welcome everybody. Thank you for joining us. This is going to be a webinar on the last gift, specifically looking at long-term survivors. This is part of a webinar series, an ongoing webinar series that is a collaboration between three of the Martin Delaney collaboratories, the DARE Collaboratory, the CRISPR for Cure Collaboratory, and the RID HIV Collaboratory. And this is a CAB initiative, community initiated and implemented. And special shout out to Michael Luella from the Red Cab and Linda D from the Dare Cab, and also Jeff Barry from the CRISPR for Cure Cab, who have been kind of the three musketeers who've helped shepherd this process. So this began last year in 2023. We had a 7-series webinar series rather on issues regarding long-term survivors. And we wanted to do one on the last skip study, and you'll learn why this is going to be so important to the long-term survivor community and especially to the cure research effort. So without further, so we're being recorded and this will be made available after the call. If you have any questions, please post them in the chat during it. Michael or I as moderators will ask those at the, of the moderator during, if appropriate, during their talks. If not, we'll save those for the end. And we do have an hour and a half for the call, so we'll have plenty of time for questions and Q&A, and perhaps ask them questions of you of the audience as well. So without further ado, I'd like to introduce Dr. Sara Gianella, and who is kind of the head of this and is going to tell us the story of Last Gift. So Dr. Gianella, welcome and take it away.
Well, thank you so much, Jeff, and thank you everyone for being here. I am really impressed about the magnitude of this event, and it's incredible to see how many people show up. And I've been working both with Jeff and with Michael for a long time. They're both great friends, wonderful people, and so I am very honored to be here, especially to talk about my favorite study, The Last Gift, who has been going on since 2017 at UC San Diego. And I think that because we work on The Last Gift, we are all better people and better researchers. So I wanted to give you a little bit of a historical background. What is The Last Gift? How we set it up? And then there are some of my colleagues. I have Dr. Robert Deiss, who is part of The Last Gift as a clinical team. And he will talk about outreach, recruitment. And then we have Dr. Karine Dubé, which I'm sure everyone knows. She is our ethicist, and she's in charge of the social behavioral part. And then we have Dr. Kate Riggs, who is also part of The Last Gift. She's one of our trainees and now soon to be faculty here at UCSD. And she will present some preliminary data. Okay. Let me start. So why did we start at The Last Gift in the first place? So as most of you know, I think all of you know, HIV is a particular virus because it established a reservoir in tissue very early during infection. And this reservoir is not only in blood, but it's really in every possible tissues around the body. So basically, we can find HIV DNA, in lymph tissue, in the gut, but also in the brain, in the heart, in the muscles, and basically every tissue that you can think about can potentially be infected or can potentially be carrying HIV. And this HIV reservoir cannot be cleared with antiretroviral therapies. And even when people are on therapy and the viral replication is completely shut down and suppressed, the HIV reservoir still persists. And we know that in the vast majority of people, when they interrupt antiretroviral therapy, the virus can come back from these tissues, right? The problem is that historically, most of the studies have been collecting blood or maybe some small biopsies because it is very hard or impossible and ethically questionable to biopsy deep tissue in living people. And the few autopsy studies that exist had some problem. And first of all, they were not following people close enough until the time of death. So often we don't have detailed information about if people were on therapy or exactly how they were doing close to the time of death. And also most autopsy study just don't perform autopsy quick enough, often 12 to 24 hours after death. And with such a long time period, we know that nucleic acid and the protein and cell viability is just getting worse over time. So this is why Dr. David Smith, who is the PI of The Last Gift, had this idea, and it was a while ago now. And we said, why are we not enrolling people that are living with HIV, and they also have another illness who is life-shortening, and they are willing and motivated to participate to HIV cure research and donate their body. And the idea was to collect blood very regularly until very close up to the time of death, and then collect the body and perform a rapid autopsy. At the time we started this, we had no idea of how complicated doing a rapid autopsy can be. It has been done just of a very small scale for cancer research, but usually for cancer, they only collect, the cancer and maybe surrounding lymph node, while here, HIV, we really like to collect every possible tissue within 6 hours from death. And so to the best of our knowledge, this is the only rapid autopsy program that was developed explicitly to enroll people with HIV. So the Last Gift study started in 2017, and the name Last Gift was really to show that this was a gift that these altruistic participants were doing to the community to try to advance HIV cure research. And our goal was to characterize the HIV reservoir across all of the tissues, and also in those people that interrupt therapies before that, because some people do, we also can see how the rebound of the virus happens and how it distributes. And our goal is to enroll 5 participants every year. So this is to give you an idea about the granularity of the clinical data that we collect. This is the time course of Last Gift 1. And you can see we collect tons of historical data, but then as we get closer to that, we have done regular blood draws. And here we basically were able to draw blood the day before this participant died. So you can imagine how precious these blood samples are, so that we can compare the virus in the blood and across the tissues. Here is to give you a little bit of an idea about how one of these autopsies looks like. We have basically between 9 and 11 people in the room, we are constantly off call. And by now we have done many autopsies. We are really a well-oiled machine. And everyone that is part of the rapid autopsy team is trained. Those are very special people. They are very committed. And we often get calls in the middle of the night, and everyone really gets together because it is our mission to be able to collect this tissue in the time frame, because everyone understands how important this is. And then usually all of these tissues, some of them get frozen, some of them get put in various media, they get shipped around the United States. Some of them, some of them go to San Francisco, North Carolina, Illinois, to Duke's University. We have so many collaborators. And then when the tissue gets there, they basically start their own process. So you can imagine how the tissue are basically non-stop worked on for maybe 24 hours until we get the best possible results. I always like to give a little bit of a human spin to our participants. And these are 2 of our participants who agreed to show their face and their names. On the left, we have Last Gift 1. His name was Tony Bennett, and he was super proud to be part of Last Gift. He always liked to joke that he wanted to become more famous of the other Tony Bennett. And you can see him here with his husband. And we actually are still in touch with Blake, his husband, because it was such a big part of Tony's, legacy to be part of The Last Gift that we remained very close. And then on the right, we have Mike Danielson. He was an incredible, funny man. He was a publicist. He spent most of his life in LA and Hollywood working with VIPs and movie stars. And so he had a very funny story to tell us. And we loved sitting down with him in his back patio and gossiping about Angelina Jolie and Denzel Washington and all these people. And both were incredible advocates of The Last Gift. Both left a huge legacy. And I don't know, I always like to tell their stories because it really shows how committed our study participants are. Then The Last Gift has been from the very beginning a community-oriented study. From day one, or even day -1, the community has been very closely involved, and Karine Dubé will talk much more about it, so I don't want to spoil anything. But really, I just wanted to say that The Last Gift wouldn't even exist without continuous commitment and advocacy from our community. And I think that we have really lead the way also to, all the social behavioral end of life. And Karine published so many papers that have really kind of opened a new field. And now a lot of people have started to show interest in this. And then we have published so far 4 high-profile biology papers. These are this one, and Katie Ricks will present some of the data, and many more are coming. In fact, if you attend CROI 2024, for those of you who are fortunate and will attend CROI 2024, we have, I think, 6 or 7 Last Gift abstract that will be presented. Okay, so these are some of the future direction that we can discuss, but I don't want to take too much time because I know we have a lot to present, and then we want to keep some time for discussion. So Jeff, what do you think? Should we move on to Karine or Robert? You're muted.
Katie is next. I am opening the slideshow now.
Okay, so Katie Ricks, she's one of our trainees. She very recently was awarded a grant who allow her to become a faculty. So I always like to tag it because they're very hard to get. So she's a real superstar. And yes, thank you, Katie, for presenting.
Everyone seeing the slides in presenter mode?
Not in presenter mode.
No, we see all of them on one screen.
Okay, here we go.
There you go.
There you go. Thanks.
So Katie, take it away.
Is Katie on the call?
You should be.
She was. I saw her.
Yeah, well, she dropped off.
There she is. But we can't hear you, Katie.
Katie, are you able to unmute?
Try again.
Can you hear me now?
Yes.
Good.
Okay, sorry about that. Can everybody hear me now?
Yes. Go ahead.
I'm going to take that as a yes.
Yeah.
Okay, apologies for that. I will just get rid of my earbuds then. So going to be a little bit of a whirlwind here. Going to go through some of the highlights and go ahead and advance. Am I able to advance?
I'm looking for your profile to make you a co-host, and then you can advance if you're sharing your slides. If not, Jeff is sharing them.
Perfect. Okay, so you can keep going. Sarah already showed some of those. Okay, so I'm gonna have two very quick intro slides just to kind of set the premise here. Basically, key things to remember are that people living with HIV are diverse and unique, and that after HIV infection or HIV is established in reservoirs, and keep in mind that it's not just identical copies of a single virus, but a population of viruses with different characteristics, fitness, different capacity to infect new cells and have replication competence, and different adaptations to react to the local immune response. And next slide. And then just to keep in mind that when we're talking about reservoirs, and this separates it in my mind too, is anatomical reservoirs, so the different actual physical locations and organ systems, the cellular reservoirs, which cells actually have HIV in them, and then the molecular reservoirs of kind of what can that HIV do, and whether it is replication competent or just able to make some viral proteins or completely silent or latent. And next slide. So going into the beginning of the last gift actual findings. So starting out was just kind of quantifying how much HIV integrated DNA is in different tissues from different samples, since this was kind of the first opportunity to really do that in kind of basically a wide variety of samples beyond the blood. And so this really started with just comparing between different anatomic sites. So down here in the lower left is just different sites within the central nervous system, so different parts of the brain, and also the thoracic spinal cord, which was, the first, also a new finding that the levels in the spinal cord, at least in these initial 6, was actually surprisingly high, and something that hadn't been recognized, and is we're fleshing that out now and getting some additional data. But I think it is important that we recognize that the different tissues have different concentrations. And then to complicate things further, as I said, everyone's unique, so it's not the same between all individuals. So on the right, we have 2 different individuals. The one on the bottom right is sort of kind of what we would expect. We anticipate that most of the HIV DNA is going to be in lymph nodes and in kind of secondary lymphoid organs like the spleen and the ileum or jejunum. And that's kind of what we really see in this person on the bottom
Jeff
right.
goes down
But
for
then
an
if you
X-ray
look
for
at the
like
top left,
5 minutes,
top
it's been
right,
like
hold
half
on.
hour. Do you know?
pancreas
Just a reminder,
really
if you're
stands
not
out.
speaking.
He's back. Oh,
And
he's here?
interestingly,
Okay.
You didn't see it.
Katie, you're muted.
I don't think she can hear, so she needs to be unmuted.
Yes, that's what I just told
you
her.
guys able to hear me?
Yes, we are.
Okay. Sorry, it just asked me to unmute. And
Yeah,
so
I had to mute you because
go
there
ahead
are
to the
people
next
on the
slide.
call that were not muted. That's all. Go ahead. You can go.
Oh. Next slide.
I think. Yeah, next slide.
Okay, and so characterizing reservoirs via sequencing. So, just instead of just measuring number of copies of DNA in a specific cell, look at the actual sequence and see how similar and how different the sequences are. And so this can tell us kind of the relationship between reservoirs. And you can click on the slide. So you can see first compartmentalization. So this is not found in every individual, but this is a particularly interesting one that does show very clear compartmentalization. So in this central nervous system and in the brain. So these gray ones here are very, very different from the rest of the genetic sequencing. And then you can also see areas of clonal expansion. So where the red box is now, which shows that all of these different colors mean that this is all the same sequence of your DNA sequence, but you can find that same sequence in multiple different samples from multiple different areas, ranging from the blood to the lymph nodes to the GI tract, etc. And so investigating further, this will show us kind of how, or to figure out those ones that we saw with the identical sequences, how did it get to all of those different areas? And so using bioinformatics and basically knowledge of genetics and virology, we're able to estimate the most likely basically source and recipient. So determining where each of those viruses transferred from. And you can go to the next slide. So this is just a preview, but next slide shows basically in several different participants, the dynamics. And so each of these, I showed the earlier one just because it was a little cleaner, but this shows, I think it's important to see how kind of messy it is and how much activity it is. And most of these people were on ART up until the end of life. Only 2 of them had stopped. And so the key things from this one is there's a lot of, most of the interaction or most of the spread and dynamics is within a specific organ system. So within the CNS or the gray areas, or within the gut or the green areas. But there's a lot of transmission and interaction between the different compartments, which is important. And also it is bidirectional. So from the CNS to the blood, from the blood to the CNS, and gut to the CNS, etc. So it's not just one way. And next slide. And then additionally, from HIV sequencing, you can also start to get a sense of how viruses adapt and expand to the local environment and possible susceptibility to cure interventions. This is based on sequencing, an estimate of susceptibility to different broadly neutralizing antibodies. So the circle in the upper right corner basically shows in each little lane is the different broadly neutralizing antibodies. And then this is all from one participant and all different sample sites. And so I think the main takeaway from this is that if you're just sampling the blood, you're not going to get, a good or accurate picture of all of the different sequences that are throughout the body, which could have impact on whether different cure interventions that are tried. And next slide. And so then moving those are more based on anatomic reservoirs, moving into the cellular reservoir of finding out which cells contain HIV. And so I think some of the most exciting, I am biased towards neuro, but some of the most exciting and groundbreaking work has been in the brain and specifically in microglia. So we've long thought that microglia we knew that there was HIV DNA and some HIV RNA expression, but not sure that, it was hard to localize that in actual microglia and to see what would happen in culture. And so this is in culture that they're able to keep isolated microglia from participants alive and show that there is integrated DNA in those microglia and RNA production. And then also Nadia Belikova is also looking more at kind of the actual subtypes of CD4 T cells that in different tissue sites where HIV is. And so you can see this is very preliminary data, but just to kind of give you a sense of, this is all CD4 cells. And so the little red dots are where HIV is detected. And you can see in liver and spleen, it really does kind of localize to memory CD4 cells, which were expected, but also there's a really, consistent finding of kind of these CD4 NKT-like cells. So has a lot more kind of cytotoxic activity than your typical CD4 cell. And you can go on to the next slide. And then also, looking kind of combining cellular and molecular reservoirs of what can the integrated HIV DNA do. So it's really important to know that if a cure intervention happens, is successful, but what if it doesn't target all of those areas? Which ones could rebound and reseed the reservoirs after taking ART off? And so this is all more of that work with the microglia. So showing that in culture, microglia exposed to latency reversal was able to have cell-free HIV RNA copies. And then they were also able to show that it was replication competent and competent for infecting other cells. And the frequency of replication competent HIV in microglia from these cell cultures was similar to what you find in peripheral circulating CD4 cells. So I think that's really important. And they were able to, just on the right, just shows that they were able to really dial down and show that it was the microglia that was producing this. And next slide. And then, so, another really important step is kind of getting a sense of where in place, in situ, are the HIV positive cells. Because are they next to lymphatic drainage? Are they directly fed by blood vessels, which is important for, pharmacodynamics of whether, interventions or medications can get to them easily, or whether those cells are actually from the blood and have infiltrated into the tissue, or whether they're tissue local or tissue resident cells. And so this is some work from Colin Kiefer that you can kind of see it. There's green arrows to help you see some of the green, but there's a few little arrows that point to little tiny dots where HIV is detected. And some of it is in microglia. It is not only next to blood vessels, which are in blue. And then you can move on to the next slide. And interestingly, he's also shown that it really is, for his work, it's highest in astrocytes, which we do know that there is HIV DNA in astrocytes and microglia, but this large difference in this high number in astrocytes was surprising and kind of really shows importance of getting a sense of where the HIV is and the interactions with the local cells and the cells next to it. So seeing where those infected cells are in relation to neurons, in relation to microglia and other structures is important. And next. I guess this is I think this is the last one I'm showing. This is other work. And so this is not actually reservoirs, but it's also important to kind of get a sense of what's happening with the HIV microenvironment and what can be happening with kind of upregulation or immune activation and different T cells and how they respond to HIV. And so this is, just the little thing on the right is just to show you this is all CD4 cells. And all of the different colors on the right are basically from different sites. And so most of them are pretty homogeneous. They all kind of group together. They're very similar in characteristics, except for this little blurble with the arrow, which is mostly made up of these kind of unique CD4 cells or have in the gut and male reproductive tract. And so Ashley George up at Gladstone and UCSF was able to show that they have some very unique viral sensors and kind of restriction mechanisms that are basically always on guard and trying to fight different viruses, but also HIV. And then next, I think, so that was a bit of a whirlwind. I don't know if we have a little bit of time for questions, but this is very similar to what Sarah had kind of already said, but our ongoing work is sequencing new tissue compartments and increasing the sample size. and then looking more at that microenvironment, so kind of the immune environment and pharmacologic environment, and then going on to identify signatures of HIV positive cells that don't require sequencing each and every cell. Okay, and I can open the chat now too, but let me know if there are questions.
Yes, there were lots of questions coming up, but one of the ones that I wanted to start with was a comment by Tom via about it's a real challenge to design medications that cross the blood-brain barrier to get to the virus in the brain or the CNS. Is that true?
Yes. So that is true. And others may be able to speak to it more too, but I think it is really important because, you know, we've kind of pointed to Timothy Ray Brown, and, you know, the people that we have had cured so far haven't necessarily had cure interventions that were targeting the brain. But I don't know that that's the case for everyone, and that's going to be, I think it is an important area that we're going to really need to consider, that just measuring in the blood for rebound and, recrudescence is not going to be enough, that we are going to have to monitor for possible reactivation at tissue level and cellular levels. But others can speak to how well we're going to be able to get into the brain.
Katie, I kind of answered most of them real time. The only one, can you explain the main cell types in the brain? I think you are more qualified than me to speak to that. So that, because they, you know, the astrocyte, microglia, neurons, like just the main cells and what they do.
Yeah. So basically neurons, you know, is what gets all the glory in the brain, because that's what has the electrical activity that kind of gets our, you know, wires our memories and everything. But then there's the glial cells, which we've rudely kind of called the support structures or support cells, but they do a lot more. So microglia in the brain are really the kind of, you can think of them as the macrophages or the immune cells of the brain. They're really important for kind of cleaning up debris and fighting infection, but also for pruning neurons to have them function their best, to form memories and things like that. And astrocytes also, we're really only now learning how much they do, but they are usually right next to neurons, and they interact very closely with both neurons and microglia, and they provide a lot of the growth hormones and the signals for neurons to grow, to spread, and also to kind of prune some of the synapses. So they're all very active, important cells in the brain that are important for our brain function.
Great, thank you. Should we go ahead and move on? I know there are lots of questions, but we're going to leave time at the end, and I think we want to keep moving. So next up is Dr. Deiss and Cheryl Dullano, who are going to talk about kind of how the whole thing works. So take it away.
Let me just say that was a wonderful talk. I mean, I really learned so much about the reservoir, some of which we've been saying to researchers, and I just can't stress enough how important it would be to have everybody see the differences in, you know, the anatomical sites and everything that you talked about. So thank you so much.
Yes, thank you.
Sounds great. Well, thank you, Jeff, for the opportunity. And it's great to see this enormous audience with such geographic diversity. And I've seen, you know, several African countries as well. So it's wonderful to be here, and thank you for the opportunity. Next slide. What I'm going to do is I'm going to share a little bit about the cohort and sort of our, there should be a slide. I think you advanced 2 slides. Can you go back one? Yes. So I'm going to share a little bit about the study cohort and, you know, who have we recruited and how we do our recruitment, because that's something that's, obviously relevant to the work we're doing. And so at this moment, we currently have enrolled 44 participants, 31 of whom have been deceased, And, on almost everyone, we've been able to perform the autopsy. So there you see our median age is 62, but we have had a range of ages as young as 30, as old as 88. 27 male, 4 female. I'll come back to this discussion of, our male-female representation. And there you see the racial demographic: 24 white, 3 black, 3 Hispanic, and 1 Asian Pacific Islander. Now, some of this reflects the sort of residential composition of San Diego County, which, in our sort of HIV demographics here, which are, do not necessarily mirror those of other cities, say Los Angeles or Atlanta, but obviously increasing the diversity and sort of mirroring the HIV epidemic in the U.S. is one of our high priorities. But this is where we're at. And participants do not always need to be enrolled right at the time of expected or anticipated death. We have had participants that outlive where we think they might be, or enroll at sort of at an earlier point when they have not necessarily an end stage diagnosis, but multiple comorbidities. So there you see the median time on our study is about 3 months. The causes of death you see there have been primarily cancer, and that's because we get a sort of a predictable time period for people's death, and it sort of is a terminal diagnosis. Oddly enough, we've had 3 participants with ALS, a neurodegenerative disease, and 4 participants with an AIDS-related diagnosis. Next slide, please. Again, you skipped back one, please. Thank you. Not sure how this happened. Okay, so this is our living participants. We have 13 currently, who are between 44 and 94 years old, actually. The median age, again, is 68. That should be 10 white, non-Hispanic individuals. Again, sort of a preponderance of white individuals in our study. And this is a group who is sort of enrolled at maybe a earlier time in their life, what we call sometimes on deck. They might not necessarily be imminent in terms of their anticipated passing, and the median time they've had in the study is 1.5 years. There again are the sort of qualifying diagnoses, and in this group, as you see, it's end stage organ disease. Is sort of the most common qualifying condition. Thank you. Let's go to the next slide. And then the next one. Okay, sorry. I think we're back to where we need to be. So this is how we recruit participants. We do sort of outreach to the community. We have a website with a pretty nice video, and we can share that website with everyone here. We provide, brochures to some of our partner organizations. One of the ways we're trying to increase diversity in our study is by performing outreach to different areas of San Diego County, such as, near the border, such as organizations that serve a predominant clientele of women. We get referrals from our parent study, which I won't get into, but it's sort of a related study to ours of organ donation. We also do active screening of our hospital census. I'm a UCSD infectious disease physician, and patients will be admitted with HIV and various conditions, and so we can see who's maybe being hospitalized repeatedly and might be approaching this period where we'd be discussing end of life. We also do database queries to see who's been referred, to find anyone in the UCSD system who's been referred to palliative care. Next slide, please. Okay, so what we did was we wanted to see how we're recruiting our participants. And so we sort of took a 1-year snapshot, identified 28 potential candidates between that period of September to August 2023. And of those 28, 9 participants enrolled. 6 who enrolled had discussed the study first with their primary care physician or oncologist rather than a member of the study team. 2 were successfully recruited in the hospital, without having spoken to their primary care provider or oncologist, and 1 patient self-referred via the website. Next, and 2 of these 9 have undergone autopsy already. Next slide, please. So who are the people that did not enroll? So among the 19 patients who didn't enroll in the study, 4 had a primary care provider, it's often myself, who was a study investigator. Therefore, we didn't sort of look at what's going on with those individuals, but we did take a look at the 15 individuals who didn't enroll to see if we could learn anything about that. Next slide, please. So of the people who enrolled or who did not enroll, there were 3 who declined enrollment during a discussion with their primary provider. Of the others, our providers often told us there wasn't either insufficient time to mention the study, or that the patient wasn't ready to discuss sort of end-of-life considerations, or might not have been appropriate based on their current medical condition. For instance, someone who's receiving chemotherapy. Time is an issue, and providers might not have time to discuss the study, or someone might feel if that a patient of theirs might not have a stable enough situation, either housing-wise, or they have a severe substance use disorder, and the provider identifies that the individual may not be an ideal study candidate. Next slide, please. So who does the initial approach? When it's the primary care provider or member of the care team, 6 of the 12 individuals have enrolled. Advance, please. And when it's the study team enrolling, only 3 of the 11. So what I really want to emphasize here is that when it's a provider that the patient already knows, or they're either their primary or their oncologist or someone along those lines, those are the referrals that we get that often tend to be successful. When it's our study team sort of finding people and talking to them about the study, it tends to be less successful. Next slide, please, or advance. There were 5 patients who didn't actually have an opportunity to discuss the study. They were either too ill at the time when we would have considered them. Next slide, please. So where do we go from here? What did we learn? Really is what I just mentioned in the last slide, that when an individual discusses the study with their primary care provider or a member of their care team, that's when the most frequent and successful discussions are had with respect to enrollment. I think in our experience, the outpatient setting is preferable to the inpatient, just because there's more time, less chaos, less stress. In the outpatient setting, these are things that tend to be sort of being considered in a planning and ahead of time, rather than, sort of right at the last minute. And that third point, as you noticed when I was talking about the recruitment methods, we're really doing a lot of active screening and enrollment, really, and a lot of that is to increase our diversity within the cohort. Fortunately, some good news, 3 of our last 5 enrolled participants have been Black, 2 of them being female, and we've recently enrolled 3 of those last 5 have also been women. So I think that's all I'd like to say. It's an overview of sort of our study and demographics and how we recruit. A lot of it tends to be sort of, I'm a physician in one of our, HIV primary care clinics, and sort of have an active pulse on this. So, we're tending to sort of do things that way through, outreach to the community, but a lot of, clinical sort of, identification as well. I don't know if there's anything left over in the chat that hasn't been answered, but
there was one question about starting ART, and do you have any idea of how long it was a time period that people had acquired HIV before they started ART?
Yes, offhand, I can get that information. It does tend to be years. Most of the participants have been living with HIV for some time. I think that tends to speak to the sort of, again, altruistic nature of the sort of study and our participants wanting to contribute something, and we've had very few people be off of ART around the time of end of life.
And Khadijah had a question too. Can you share some
Yeah.
of the concerns that were raised by potential participants that needed to be resolved before enrolling?
Yeah, we have Cheryl Dullano, who I want to, sort of recognize as being one of the persons who, does the actual enrollments and talks to participants. I know one thing that keeps us from enrolling sometimes is someone's family might not be aware of their diagnosis. And, this tends to be a program where people will need, family support and understanding, especially notification of illness and death. So, that's sometimes a barrier for individuals. Cheryl, do you have any other thoughts on, some of the things, our participants are concerned about before they enroll? I thought I saw.
You're muted. Yeah, we can't hear. At least I can't hear you.
And I thought I just saw Cheryl in the list of participants at some point, but if not,
She is here. She
There
was
she is.
She was talking, but we can't hear her. I saw her
see.
moving her lips.
There she is. Yes. I think, I just wanted to recognize her work. I unfortunately can't hear because she does a great job, communicating with our participants. What I will say often comes up are sort of logistics, and cremation and how to get the remains. We are able to do open casket for people, but usually tend to do cremation. So I think logistics tends to be sort of a big one, and people want to know like what happens, sort of some of the study findings, and so that we're able to share some of that material that we've just had, and those would probably be the big ones, I think, yeah.
I know Carol has re-entered, Cheryl, rather. Have you been able, Cheryl, are you able to unmute? And
Can you hear me now?
yes, we can. Terrific. Thank
Okay.
you.
Hello, everybody. My name is Cheryl Dullano, and I have the distinct privilege to be able to enroll these participants on the last gift study. Once they have said to whether it's their primary care provider, yes, I'm interested in hearing more. So the phone call is very interesting, as you can imagine. But I will say not so much a barrier, but I think one of the things that has to be stressed to the participant and their next of kin is, yes, once their loved one passes away, there's sort of this 6-hour timer that's going on in the background. And we want to make sure that we respect the next of kin in terms of, being able or given the proper amount of time to say their goodbyes. That said, I also want to bring up something that's very important, and Dr. Karine Dubé will speak more about that with her fancy graphs and everything, but I also do the social behavioral. I'm sorry?
Go ahead.
Okay. I also do the social behavioral interviews with the participants and their next of kin. There have been some, sort of community board members who want to make sure, are you establishing sort of this respectful relationship with the participants and their next of kin? And the answer to that is yes. We recognize, especially in this sort of research, that the next of kin plays a very pivotal role. We depend on them to keep us informed, particularly in the unfortunate event of the participants passing, as they do serve as our primary contact to notify us of any changes if the participant status. And so because of that, not only do I make it a point to develop these relationships with our participants, but actually their next of kin. And so in doing these interviews, they are recorded. And again, Dr. Karine Dubé will speak more to them. But we want the participants and next of kin to understand that we are always interested in hearing, what their insights are, especially once their loved one has passed. We make it a priority to ask them, is there anything we could have done different or better? And I will say that a recurring theme among the next of kin is their appreciation for the study, and that it actually has sort of deepened their connection with their loved one, because not only is their loved one contributing sort of to science, but they also feel in, having this relationship with the study team that they are also doing the same.
Yeah, thank you, Cheryl. I know Sarah put it in the chat, but Cheryl really is the heart and soul of the study, being the one who interacts the most with the participants, enrolls them, as she said, and also their families on an ongoing basis, and even after the participants have passed. And so she and Susana Concha Garcia before her have really kind of set the tone for the study, and how empathetic they are and sensitive to everything. And we couldn't do it without them, because as you can imagine, it's a fraught time of life. There's a lot going on, and they really kind of pull the whole thing together for everyone, including the study team. So thank you
Jeff,
for the work you do.
since I've seen many questions about if people can donate if they are not in San Diego. So usually we require people to be in San Diego, because obviously we need to be able to collect the body to do the rapid autopsy. We have occasionally accepted people that are a little bit far away. We had one participant in LA. We have been, always discussing about Palm Springs. It's a little bit of a case-by-case decision, depending on, also the next of kin commitment, and if if we're all on the same page and we can make everything happen very fast. So I'm not 100%, Orange County, Riverside, as long as it's like a car ride away, and we are all, and the communication is very good, and the participant is very committed. We have done it in the past, but it's a little bit of a case-by-case decision, just to make sure that, because it will be very sad if then we cannot execute the autopsy because there is traffic or the next of kin doesn't call us. So reach out, and if you have a question, reach out, and we can look at every case individually.
There's also our parent study, which is, sort of 24 hours, and, is focused on the brain and central nervous system only. And there are sort of partners throughout the country as well, I believe UCLA, but others. So reach out to us. We're happy to sort of talk things over, to a greater extent.
And I know a lot of people who aren't in Southern California have been asking about this, and Nina Martinez has been sharing some really great information on other networks for organ donation and body donation as well. So thank you, Nina, for that. So Cheryl's kind of set the stage for Karine Dubé. So let's segue on to her. Karine, take it away.
Thank you so much, Jeff. As you all can see, The Last Gift is a wonderful program that not only has really robust basic sciences and an amazing clinical team, but it is the only research program that I know of that really elevates social behavioral sciences, ethics, and community engaged research to the same level of the basic sciences. So it's really quite unique. And we employ a 360-degree approach to these buckets. We see them as interrelated. They're distinct, but also they relate with one another. And we'll talk about why this is important. So next slide. So Dr. Gianella has already showed how we've been able to engage the community. This was a special issue in the Positively Aware About the Last Gift, where we talked about the terminal kindness of the early participants. Next slide. And we've done interviews with each of the Last Gift participants if they are willing to participate in the program. So they do a very thorough baseline interview, and then they do follow-up interviews depending on their prognosis. So we found that people find a deep sense of self-actualization being part of the Last Gift. They appreciate being able to leave a legacy, have hope for research, appreciate compassion, and also really want to make sure that their bodies are treated with dignity at the end of life and after the end of life. Next slide. In terms of the next of kin and loved ones and intimate partners, we also engage them into the study. only if the last gift participants are willing to refer them into the study. And the next of kin and loved ones really appreciate being included. They appreciate being supported throughout the end of life process. For some of them, it's the first time they've been having to deal with something like this. They also appreciate the mental health and emotional support they receive, and the communication and professionalism from the research team, and so on. Next slide. And then we also did focus groups with clinical and rapid research autopsy staff to make sure that the program is also acceptable to them as well. And we find that there's tremendous resilience in the team itself, and they're able to manage stress of having to deal with the end of life. They also express tremendous gratitude to the last gift participant and the interdependence between one another and with the community, and also have really deep ethical awareness. Next slide. And so we're doing social behavioral research to ensure that the study remains acceptable to the last gift participants. So this slide shows the perceived risks and benefits of the last gift program. As you can see, the perceived benefits outweigh the risk. This is important to measure to make sure that the program remains within ethical guidelines. The issue of cremation has actually generated quite a bit of an ethical debate. Even though participants may perceive cremation as a benefit, we are unable as a research team to present this as a benefit. And this is something the Community Advisory Board has actually had to advocate quite a bit with the IRB, the Institutional Review Board at UCSD, because the IRB initially said cremation was coercive when the community members said, no, this needs to be part of the study. It needs to be part of the design. It's a basic necessity. But overall, participants perceive tremendous societal benefits of being in the program, also emotional and psychosocial benefits, and mostly having somebody to speak to at the end of life, and not being isolated. And then in terms of the perceived risks, the large blood draws was the only risk perceived in the closed-ended and survey component. And then next of kin tend to be a little bit more protective, so they do perceive a little bit more risks of being part of the last gift, but overall, they've been incredibly supportive. Next slide. And then we find that participants find tremendous sense of meaning and peace of being part of the last gift program. Next slide. And this slide shows a quality of life data collected over the last 7 years. It doesn't seem like it's a lot, but it's actually every dot represents a very thorough survey about quality of life at the end of life. We are employing the Karen Steinhauser survey, and you could see the trend there. So we are going to very cautiously interpret those data, but what we could see is that quality of life at the end of life is not declining overall. And this makes us hopeful about the last gift program, that we might be doing something here. So we are very cautiously optimistic about what the last gift does at the end of life in terms of providing support. Yeah, next slide. We can only think about where we started in the early days of the HIV epidemic in terms of quality of life at the end of life, and we think we've come a really long way. This is a framework that was created by the community in collaboration with the community as participants were making requests for donations of their samples, and as we were receiving external requests for the last gift, we needed a framework to adjudicate donations of human biological materials in the context of cure-related research at the end of life. And you can see our ethical values, like respect for persons, social and scientific validity, altruism, trustworthiness, stewardship, and community input. We really believe in those values, and these must remain in balance. And Tom Villa must be credited for helping us with this framework of how we were to prioritize the sample. So we always start with directives from participants and then HIV cure research. And then if there were to be additional requests, then of course we have to have really robust informed consent. but samples could inform other fields of research. We find that participants are very altruistic, and even though their altruism is motivated by HIV, they usually want to help benefit other fields of research. Next slide. Also with Jeff Taylor and Dee Cates and the Antiviral Research CAB, really early on in the program, we created ethical considerations. This paper took over a year to write. It was a really difficult paper to write because we had to really look at the literature on ethics and HIV cure-related research and the end of life to figure out an ethical framework for the last gift. And so we came up with 5 key ethical considerations, including protecting autonomy through informed consent, avoiding exploitation and fostering altruism, maintaining a favorable benefit to risk balance, safeguarding vulnerability through patient participant centeredness, and ensuring acceptance of next of kin, loved ones, and communities. And that's why we're also opening up the last gift to a more wider conversation. And that's why we're doing the webinar today. And community patient participant centeredness really remains central to the study. So participants are being centered as part of this program. And there are many ways we center participants. Next slide. We also did a lot of community-based, actually stakeholder kind of engagement, mostly in the San Diego area, to understand perceptions of the last gifts and ethical and practical considerations. We also documented our ethics lessons learned throughout the program, including the intercurrent COVID pandemic, the dual role of clinical care and research, informed consent, communication, the logistics, legally required autopsy versus rapid research autopsy, emotional support, determination of next of kin of loved ones, boundaries. We've already talked about cremation perceived as a study benefit, and the topic of vulnerability. In fact, Dr. Gianella led a paper really challenging the notion of vulnerability. We find that participants in the last gift do not necessarily consider themselves vulnerable, even though they are at the end of life. They once face the prospect of their own mortality, and they really understand what this is and really want to help advance the science of HIV persistence. Next slide. And now our next frontier would be to look at acceptability of introducing intervention. So this is still very hypothetical at this point. The last gift is purely observational, looking at the HIV reservoir. But the next step would be, what if we were to introduce interventions? So we're looking at acceptability of these different types of interventions. And of course, these types of studies would depend on the ethical considerations, benefit to risk balance, but also on funding. But overall, there was a good acceptability of some of the interventions that may be suitable for this type of research. And we can include that in the discussion as well. So next slide. And this was the perception of next of kin and loved ones. So we also found a relatively high acceptability among next of kin and loved ones. And the next slide. And then there's also a really strong willingness to donate tissue biopsies at the end of life. Next slide. And we also looked at ethical considerations for interventional HIV cure research. This is an ongoing conversation that we are having with the community. We even had a meeting with the FDA, and there was a really interesting exchange, sort of speaking with the FDA about whether this type of research would be acceptable. Next slide. So we want to thank our UCSD Dream Team Lab. I would like to thank Hirsch Patel and Simon Duque and Whitney Tran and Fong Wang, who help us with the social behavioral component. Next slide. And maybe, Jeff, you want to talk a little bit about this, but we do have an open social behavioral research study right now focused on long-term survivors of HIV. So there's 2 components to that study. The first one is understanding the perceptions of long-term survivors and needs and priorities related to HIV cure research, not related to the end of life. And then the second interview opportunity is focused on HIV cure-related research at the end of life. And so we're currently recruiting people who are 65 years and older, have acquired HIV before the start of combination therapy in 1996, and are living in the United States or its territories and willing to provide informed consent. And we also just got IRB approval to do interviews with just the end-of-life component. So if anyone would love to give their thoughts about HIV cure-related research at the end of life, please feel free to contact either Jeff or myself. And I think that's it. Thank you so much.
Jeff, we should save questions for Karine or others till the end, right? Or what? How would you like to do that?
Well, so we are at the end, so now we can take questions.
Okay, cool.
Yeah, I just wanted to thank Karine for all she's done for the study. As this Dr. Gianella alluded, we couldn't have done this study without having her input at the very beginning as a sociobehavioral researcher to really prove to the regulators and to the IRB that this is something the community wanted, that it was not coercive, and that in fact it was offering the people an opportunity and to leave a legacy, as the last gift name suggests. So kudos to the team for realizing that early on and bringing her on, and Karine for all the amazing work she's done with the community and the study participants to really document this in a scientific manner going forward. I think it's really groundbreaking and kind of sets the tone for how we should be doing this in other areas of cure research. So, Michael, I know you've been trying to keep up with all the questions popping up in the chat. I know some people have been answering them in the chat, but if you want to...
Right. And so one of the questions I had, it was talked about, this is very vague. You're talking about end of life intervention studies. What are you talking about with that? What does that mean?
Yeah, good question. Maybe Robert could answer that.
Robert, you're
I'll
muted.
do my best. This is something sort of, that we're at the nascent stages of developing and thinking about. Right now, the last gift study, everything that we've been talking about has been descriptive, right? So finding, the virus in certain reservoirs. We're seeing how it interacts with the host, et cetera, host tissues, et cetera. What we'd love to do is, to see what can be done in terms of therapeutics, let's say, right? We have someone maybe who is enrolled in our study, agrees, has, let's say, been given a prognosis of 3 to 6 months, and maybe that individual would consider a treatment interruption, right? To be off ART, so that we could then, sort of be analyzing blood and, primarily blood, but maybe other tissues along the way in the pre-mortem period. But then at the time of death, autopsy, we could see sort of what's happened. Someone who would receive a treatment interruption, obviously, we could look at new therapeutics and see how those are working, and be able to analyze those along the way. And, sort of at the autopsy, what happened to the reservoir? Did the sort of candidate drug reach all the areas we wanted it to, we needed it to? So, there's a lot of studies like this among living participants, right? Where people undergo a treatment interruption to investigate a new drug. The unique thing about our, what we eventually will propose and what we think is going to take last gift into the future, are sort of those similar treatment interruption studies looking at a new drug and what happens, in that pre-mortem and post-mortem period.
Linda, you have your hand up for a question.
Yeah. At first I wanted to say to Dave that when we had our call with the FDA, or actually it was in pre-discussions to the call, I mean, there was some hesitancy about interventions that might be too risky, even if somebody was near death. So, I mean, it's the first time they've actually heard that or encountered that. So it might be something that with some time we could, finesse. But initially it was interesting that they were like, wow, doesn't matter what stage of life you're at, that it may be that the risk benefit ratio remains the same with everybody who's still walking and talking. So it was interesting. But anyway, my questions were for Karine. Very quickly, the first one was, in the beginning of your talk, you said, you showed some evidence for people having I guess I'm not pushing it to say that maybe the same quality of life or a better quality of life than most people that are near death. And that there's a tendency to make people depressed and to lose hope and stuff like that. I mean, I'm not sure what you need to do to prove that in a larger group, but I mean, I think that's really important to show exactly the benefit that somebody gets in relation to what I was just saying to Dave, but just generally. And oh yeah, the other thing, Karine, I mean, back to the FDA, do you want another meeting with them? Were you happy with the people that we got there this time? I mean, I thought that ethicist was a little general and wasn't familiar with this kind of stuff, but what would you like to do next in that regard?
Well, thank you, Linda, for those comments. It's the quality of life data are descriptive, so it's really hard to compare because we do not have a control group, but there's some evidence there that quality of life is being preserved. And we also think that the quality of life is contingent on the determinants of health, and we're more actively incorporating that as well into the design. In terms of the FDA, I think this would depend on the unique intervention, if there were to be one in the future, and on the funding. So it's really hard to predict at this moment what intervention would be a prime candidate. But we've heard from the community, I think Jeff has heard it, I've heard it, that people really want their contribution to count and to really matter in helping advance the field. And we can learn a lot from observational study. The interventions would really help propel the science, particularly if you were to think about like a cell and gene therapy intervention, like a CRISPR or a CAR, or, and so I think this would depend on funding. Like we need to write grant. The LAC gift is renewed every 5 years. So we need to be writing grants, and then let's see. And then I think once we have a potential candidate, it would be nice to re-engage with the FDA. In our study PI, Dr. Davis Smith, of course, should be included, and Dr. Gianella and community as well as part of that conversation. But I think the November 6th meeting, we had a private counseling with the FDA, and I think that was a really great first step. Thank you for that, for your help with that, Linda.
Mark, I see your hands up.
Yeah, I wanted to kind of chime in, being part of the MaxWise combined cohort study since it started in '83, and I actually serum converted with HIV between 84 and 85. They did have an autopsy program, but it ended about 6 years ago. We're hoping to get that resurrected, but, the last gift project is so vital to understanding more about the reservoir. So yeah, as you've spoken, it is, truly the final gift that we can give until there's a cure, about understanding the reservoirs. And the last gift project, does their autopsies even better than what the MaxWise combined cohort did, because they did it in a much more timely fashion, so that everything is preserved. So the science is going to be top notch that will come from that. So, it's vital. And I hope that, NIH sees that and funds it. And, this project goes until we no longer need it. But, scientists need to have access to the tissues that will gain understanding that will help us get to a cure. So I just wanted to throw that in from my perspective as a person living with HIV, as well as a scientist taking advantage of tissues that we use to study the reservoirs as well.
Thank you, Mark.
Thank you.
We also have an interesting question here. Someone was looking for some clarification on, does this study provide cremation costs or burial costs for participants who could benefit from that?
Cheryl can answer, but
I'll allow you to go first, Karine.
cremation is included, but we cannot portray as a benefit of the study for ethical reasons.
Okay. Hopefully that
Sure.
answers.
Could you maybe expand on kind of how those conversations go when you're having that with participants and their next of kin when you first approach them?
So that is part of it, is written into the consent that if they would like for the some people have sort of prearranged, whatever it is. But something that we do say, and again, it is in the consent, it's stated that if that is something that's not already been prearranged, that the study, the last gift study, will cover the cost of cremation. And I think something to mention, and that this is something that I've also never thought about, is typically when people donate sort of their body to science, their family or their cremains rather do not go back to their family or their next of kin loved one. Whereas with the last gift, the family they will receive their loved one or the cremains, so that they can do sort of like a service or anything like that if that's what they choose to do.
Thank you. Also, we have another question that's popped up a few times from Reggie, who's wanting to know about how this information from this study, from this research, could best benefit our community.
Who wants to take that one on?
How about Dr. Riggs? If I may pick on someone who's, sort of this group most in key with the findings. I think, on a general level, obviously, the research that we're doing, we're trying to uncover as many, discoveries and advance the field in that manner. Katie, maybe you have some specific thoughts.
Sure. Yeah, I think, everything that's been talked about has different implications. So, what Karine and Cheryl and everyone has described of just understanding the landscape and what is needed for going forward with end-of-life research and how to do it ethically has been, a very unprecedented and monumental impact, showing us a way forward. And then from the kind of research and more, lab science standpoint, I think it's really shown us, my key takeaways are that we do have, we've scratched we've seen the tip of the iceberg, and we have a lot more work to do. But I think the takeaways are it is complex, but we are every day understanding more and more about the diversity between individuals and the diversity between different sites where we have reservoirs. And I think it's really important for us going forward in cure research to make sure that we all advocate for kind of monitoring for all of those different sites and monitoring for adverse events. So we do this safely and monitoring for efficacy. So I'm not sure if that answers it, but that's kind of what I think.
If I see the comment from Nina in the chat about let's see, and Reggie again, attempts at being made to diversify study subjects. Yes, thank you. So I mentioned in sort of my segment that we're doing a lot more sort of active recruitment and case finding. We'd presented, and sort of most of the providers in San Diego County, we hope, are aware of the study at this point. And so, by sort of analyzing individuals who come through the UCSD system, we are the referral center for, many sort of individuals who have, tertiary illnesses or cancers or require specialized care. So we do find we have maybe more access than other health centers might, and we're the safety net population or sort of setting for our hospital where we tend to operate out of is sort of one of the safety net hospitals for the region. And so in terms of finding a population who might not be, looking actively on research and website, that's how we've tried to expand and diversify the pool, sort of finding people where they are rather than them coming to us, if that makes sense. There was a question about veterans as well, and one of our hospitals and several of our study investigators and providers are physicians at the VA hospital. So we have had individuals referred from the VA who have participated in the study. We also sort of have done our presentations at the Naval Medical Center where I used to work. And so, that's sort of more active duty population, obviously, but we have many close collaborators at that institution as well.
There's one other quick question in the chat that I wanted to make sure was asked. Someone was asking if the tissue samples that are collected are able to be shared with collaborators outside the US.
Yes, there is a concept sheet concept, and we use the prioritization framework that we showed earlier.
Okay.
Every request is vetted, and then if we need to go back to the IRB, we go back to the IRB.
Yeah,
Great.
Karin, that came from me. It's Linus here because we haven't been participating yet. And I think, you know, the number of people that do in-depth reservoir research, although the number of labs is expanding, it's still somehow limited. People that do integration sites, full length, and so on. So maybe it's good to think about a network that also cost and resources are a bit more shared. We could think about that. I just bring it to the table here, but I'm open to discuss later. Thank you.
Mm.
Yes, and the last gift is part of several of the Martin Delaney collaboratories as well. And samples are being shipped with, well, they're in San Diego, but also across the United States and even internationally as well. So the last gift is very collaborative. And I think that talks about the social value of the research. Even though right now the program is in San Diego, there's researchers across the country who can learn from those samples. And we have several conversations about what it means to work with those extremely precious samples. And that needs to be part of the conversation as well. But in terms of the community input, we would love your thoughts. We would love to hear from the community about what you think about this type of research, because is this something that should be expanded to other sites? And we would love to hear your thoughts. And also, if you think that if you would like to reach out to any one of us, we're happy to hear your feedback about the program. And I do want to say that somebody asked about we prefer to call participants and volunteers to acknowledge the active participation in the last gift. So we are very strict with language in the program, and we use person first language in as much as we can. So thank you all so much.
Mm-hmm. So we're getting close to the quitting time, and I see Linda has her hand up, and so does Karine. So let's go to Linda first.
Okay, a couple of things. I mean, I think, you know, it's really the work that you've even shown today about the differences in compartments and in, you know, is so essential. I mean, I think a lot of researchers don't look at it that way. I mean, they kind of think about I don't know what they think about, but they don't think about this. And if you could show if there was some sort of a host factor, which we've been trying to get them to look, many researchers to look at for years, and also that, you know, there are these differences. I mean, that's essential to research if we're going to ever have anything that's going to work for different people. You know, or, you know, there's been so many studies where something works somewhat in half, but not the other half. So, I mean, identifying what's up with that is just essential, as I said, the different characteristics of the reservoir in different parts of the body. The other thing somebody was mentioning about how important this is, and I'm sure that there would be many organizations, including people on this call, works from people on this call that would be happy to write you letters of support for any other grant that you might file.
That's wonderful. We appreciate that thought. And you know, and the comments as well. And I'm reading many in the chat. And so thank you all for attending, you know, and for commenting and being a very active audience. And we benefit from, you know, every time we present these to whatever audience, comprised of whatever sort of you know, community, we always benefit from hearing from individuals. And so thank you, just as Karine says, who always says things more eloquently than I do, for being part of the conversation. And I'll also just thank Jeff while I have the floor for the invitation to be part of this hour and a half, and for getting our slides together and helping with that, and to Michael as well for coordinating and leading things on this end.
Terrific. Well, thank you all, everyone, for attending. We've had an amazing turnout. I think at our peak it was about 120 people, and some amazing, questions and feedback. A lot of really great interaction, which is really what this study has been about from the beginning, as I said before. And it's been a real privilege and an honor as a community person to be brought on at the beginning to help kind of advocate for the experience of long-term survivors and why we want this. And working with Karine to find a way to put that in scientific terms that would be accepted by the regulators and IRBs, so that this could actually happen. So more to come. As you can see, there's a lot of interest from all over the world in this. We would love to see this replicated elsewhere, as we've been seeing in the chat. And as our researchers at other Delaney collaboratories, I'll put a plug for the Martin Delaney collaboratories. There are 10 of them now, and they can all use these data if they're interested in the tissue samples. So, please reach out to us, and hopefully we'll see this replicated in other places as well. I know other places are looking at it. So we've kudos to the team for kind of setting the stage for making all this happen, and all of you for joining us today. So this has been recorded. They'll be recording. Michael will work his magic, and we'll be sending the link out to everyone who was registered for the call, and posting it on the RID website, as well as the various Delaney collaboratory websites. And stay tuned. We are going to be having in 2024 another series of webinars focusing on the reservoirs. So this is a good kind of intro into that, and how we measure them, and how important that is to cure research. So stay tuned for that, and more to come. So thank you, everyone. And folks in the East, please stay warm, don't freeze.